This immune systems overview is presented by Brian Geniuson, founder and research systems architect at Natural Scientifics and research scientist in bioactive and systems biology. This analysis examines immune physiology through a preventative lens, focusing on molecular, cellular, and metabolic signaling that support immune resilience, balance, and regulatory control, rather than disease treatment or clinical intervention. At the molecular level, immune function is governed by coordinated signaling across innate and adaptive immune cells, including T lymphocytes, B lymphocytes, natural killer cells, and antigen-presenting cells. These networks rely on tightly regulated pathways involving cytokine signaling, metabolic reprogramming, redox balance, and energy availability to maintain appropriate immune responsiveness. Immunometabolic regulation plays a central role in immune adaptation. Signaling pathways involving mechanistic target of rapamycin, AMP, activated protein kinase, glycolytic flux, mitochondrial oxidative phosphorylation, and reactive oxygen species act as contextual regulators of immune cell activation, differentiation, and resolution. Cellular defense mechanisms include phagocytosis, complement activation, antibody-mediated neutralization, and extracellular trap formation. These processes contribute to immune surveillance and threat containment while requiring precise regulation to prevent excessive inflammatory signaling. Immune tolerance and regulatory control are maintained through anti-inflammatory cytokine networks and regulatory immune cell populations, including regulatory T cells and interleukin-mediated signaling pathways. These systems support immune equilibrium and limit chronic inflammatory burden. In human research, immune-related biomarkers such as inflammatory mediators, immune cell distributions, and metabolic indicators have been evaluated across diverse study designs. Findings are heterogeneous and influenced by baseline immune status, metabolic health, age, environmental exposure, and methodological variability. Importantly, modulation of immune signaling pathways or surrogate biomarkers does not equate to immune disease prevention or therapeutic benefit. Translational limitations apply, and physiological immune responses may not produce clinically meaningful outcomes at the individual or population level. This content is provided for educational and research purposes only and does not constitute medical advice. Always consult a qualified healthcare professional before making decisions related to supplements, medications, or health interventions. Related molecular and systems biology research is available across the accompanying videos and playlists.